Good morning, everyone. Thanks for attending today's seminar. It is my pleasure to introduce you to Lisieux Fuzessy, also known as Lika. I'm going to say some words about her trajectory. So Lika, or Lisieux, is a Brazilian ecologist with a strong background in community ecology, evolutionary biology, and conservation. She holds a PhD in plant biology from the Federal University of Minas Gerais in Brazil, with research focused on vertebrate-mediated seed dispersal in neotropical ecosystems. Lisieux has held postdoctoral positions at UNESP in São Paulo, Brazil, including a one-year research stay at the Estación Biológica de Doñana, where we met some years ago. And then she was later a Marie Sklodowska-Curie Fellow at CREAF in Catalonia, where she's currently based with a short stay Severo Ochoa mobility grant. Her current research explores how human-driven changes reshape ecological interactions, biodiversity, and ecosystem functioning. So thank you, Lisieux.
Hello, everyone. Good morning. First of all, I'd like to thank Anna for the invitation. It's a pleasure for being here, and thank all of you as well for being here to attend the seminar. And I'm here today to present the talk Between Legacy and Flexibility, Drivers of Mutualistic Interaction Patterns in a Changing World. And this is work that I've been done for the past few years of my research. And I will present some questions that I have, some questions that I already answered, and some still another things that I have in mind. So at the end, I'll be glad to answer questions and also discuss new ideas with all of you. So as most of you might know, ecological interactions are very important for the maintenance of biodiversity in tropical ecosystems. Mutualistic interactions, such as Frugivory, helps us to determine how ecosystems assemble, resist disturbances, and also function. Despite their great importance, tropical ecosystems are under threat by human intervention and also environmental change. So a central question in conservation biology today is how are interactions structured in tropical ecosystems and how do they respond to change. Something that it's very important to consider and to have in mind before I say anything is that network structure is not a static thing. So it's all the time being shaped by a dynamic interplay between ecological filters, evolutionary legacies, and also the potential for interactions to reorganize. So to answer this main question of how are interactions structured in tropical ecosystems and how do they respond to change, I've been using two distinct perspectives where on the first perspective, I look at the evolutionary legacy, which means that how much of the shared evolutionary history between interacting clades determine the structure of interaction patterns that we see today. So I ask how much the shared history dictates who interacts with whom. And on the second perspective, I move to the ecological responses and I ask how do networks reorganize when partners are lost. So for the first perspective, the main aim is to assess the weight of evolutionary history in shaping interaction patterns. So for this, I test the hypothesis, if evolution is the main driver of interaction structure, we should see a strong cophylogenetic signal. But what is the cophylogenetic signal? The cophylogenetic signal provides us insights into how interacting clades have influenced each other over time. So when the phylogenetic trees of both interacting clades show a similar branching pattern, it is an indication that interacting groups have evolved in a correlated manner. So when those interactions among clades happen over macroevolutionary scales, changes in one clade may trigger evolutionary responses in the other clade. However, it is very important to keep in mind, and this is really important to consider that the presence of cophylogenetic signal does not mean the presence of coevolution. Those are two distinct processes. Mainly because coevolution implies reciprocal effects in each other's evolution over time, forming something known as a coevolution spiral, where no? Ahora? Sí? Vale. Changes in one clade trigger evolutionary responses on the second clade, and those responses trigger evolutionary responses in the first clade again, and then forming like something like a coevolution spiral. On the other hand, cophylogenetic signal only indicates that phylogenies of interacting clades, here in my case, phylogenies of plants and phylogenies of vertebrates, are congruent in some degree. This degree of congruence might be driven by two main groups of processes: ecological pressures, such as phylogenetic tracking, host shift selection, and evolutionary conserved traits, and also biogeographical events, such as vicariance. In the case of phylogenetic tracking and host shift selection, this congruency between the two phylogenies is high. So every time that we see a high congruence between the two phylogenies, we might conclude that this pattern is being driven by either phylogenetic tracking or host shift selection. In the case of evolutionary conserved traits and vicariance, the degree of congruence may be variable. It does not mean that the congruence has to be high to consider that some ecological pressures or biogeographical events are affecting the evolution of both clades. For the past 2 decades, many tools have been, we have many tools to test for the presence of the cophylogenetic signal. And one of the most important tools are PACO, the Procrustean approach to cophylogeny, that help us to assess how well phylogenies of interacting clades align in terms of branch lengths, topology, and also the overall fit. Basically, PACO tests the null hypothesis that there is no significant cophylogenetic signal between the two phylogenies, which means that PACO tests whether closely related vertebrates tend to interact with closely related plants at the same time that closely related plants interact with closely related vertebrates. As I said before, this might generate a high degree of cophylogenetic signal, which means high degrees of phylogenetic congruence. It might generate low degrees of cophylogenetic signal, which means low degrees of phylogenetic congruence, and either high or low degrees of cophylogenetic signal, meaning that past evolutionary history has somehow shaped present-day interaction patterns. If we see a high level of cophylogenetic signal, means that evolutionary history has a great importance in shaping nowadays interactions. But if we see a low cophylogenetic signal, it does not mean that evolutionary history is not important. It is there, but in a degree a little bit lower. So PACO uses this Procrustean superimposition to align distance matrices that are derived from the phylogenetic trees. So for example, here we have the plant phylogenetic tree, and then from this phylogenetic tree, we derive a distance matrix based on phylogenetic distances, and then a phylogenetic tree of the monkey or of the vertebrate, and then we derive a phylogenetic distance matrix. And then PACO makes the superimposition between this matrix with this other matrix. And it minimizes the differences between the two configurations and gives us a value of congruence, which is the sum of the squared residuals between the two matrices. A great advantage of using PACO is that this superimposition gives us an individual value of contribution to the overall cophylogenetic signal, which means that for each interaction, we have a value of contribution to the overall cophylogenetic signals. So it allows us to test for individual effects, for group effects, and also to add trait values or attribute values into our analysis. So a significant cophylogenetic signal, either if it is high or low, means that somehow past evolutionary history has contributed to shape present-day interaction patterns. For the past few years, I've been testing for the presence of cophylogenetic signal in vertebrate-plant interactions in tropics. And the first work that we had, we tested for the cophylogenetic signal in interactions between fruits and frugivores and monkeys in the whole Neotropic. And what we find is that there is a significant but with low-level cophylogenetic signal in the case of neotropical primates and fruiting plants in neotropics. When I moved to Madagascar, I also tested for the presence of cophylogenetic signal in mutualistic and also in antagonistic interactions. So I tested for the presence of cophylogenetic signal in seed dispersal and also on seed predation, folivory, and also in florivory. And what I find is that there is a significant, although low cophylogenetic signal in all interactions, either if it is antagonistic or if it is mutualistic. So the significance of this signal means that closely related plants or primates and closely related lemurs interact with closely related plants. So somehow past evolutionary history is acting and contributing to shape present-day interaction patterns in neotropics and also in Madagascar. However, the low level of cophylogenetic signal indicates that we have a weak congruence between the two phylogenies. So this means that phylogenies between primates and plants, neotropical primates and plants, and lemurs and plants do not have a very straight match. So as I said before, some degree of congruence might be driven either by ecological pressures, phylogenetic tracking, host shift selection, and conserved traits, or by biogeographical events such as vicariance. It is very unlikely that phylogenetic tracking and host shift selection are the best explanations for what we are observing here, since as I said before, when phylogenetic tracking and host shift selection are operating, we tend to observe high degrees of cophylogenetic signals, so it should not be the best explanation here. And looking at vicariance, it is very unlikely to be the best explanation as well, because when we check for the presence of cophylogenetic signal, including the whole neotropics, we observe a pattern, and then the same pattern is replicated when we share the analysis into the three main moist forests that we have in neotropics, which are Atlantic forest, Amazon, and Mesoamerican forests. So it is very unlikely that vicariance is playing an important role here. Also, moving to Madagascar, it is very unlikely that within a relatively small island, vicariance has a great importance. So something that is very important and that we need to take into account is that vertebrate-plant interactions are very complex and involve multiple actors. Because until here, I was testing the presence of the cophylogenetic signal only using primates and lemurs as my objectives, my individuals. But we need to consider that vertebrate-plant interactions, it is very unlikely that a fruit is consumed exclusively by one species or one group of vertebrate. Also, vertebrate and plant interactions lie in a continuum between mutualism and antagonism. It is also very unlikely that one individual or one species acts 100% as a seed predator or 100% as a seed disperser, and this role plays different pressures in the final outcome, that is evolution. So for the past 2 years of my research, I made a step forward and also test for the presence of cophylogenetic signal in interactions, including bats, birds, and primates, which are the main seed dispersers and frugivores that we have in the neotropics. Not surprisingly, when testing for each of these groups, we find a significant signal, but it's still low level. Which means that evolutionary history is detectable, but might not be the dominant force determining network structure in neotropics. Even when we filtered the interactions for the plant families known to be strictly related to each vertebrate group, for example here, Sapotaceae with primates, Melastomataceae with birds, and Piperaceae with bats, we see that the intensity of the cophylogenetic signal increases a little bit, but it remains too weak to be the best explanation for what we observed as interaction patterns nowadays. So the conclusion until here is that specialized interactions do carry a stronger but still weak coevolutionary signature. So it made us think, should functional attributes of vertebrates and plants determine the strength of the cophylogenetic signal? So as a second step forward, we tested for the influence of attributes from plants and vertebrates on the strength of the cophylogenetic signal. As I said before, this is something that Paco allows us to do since we have a value of contribution for each interaction. So we can isolate the amount of contribution based on, for example, traits, what we are doing here. So for vertebrates, we use the diet, which means how much of each item in the diet those animals have. Body size, which determines how much they can eat, and the individual network role as measures of centrality, which are species strength and specialization. For plants, we selected traits that we know that are related to the preference or the selection by vertebrates, which are color, fruit type, husk presence. Husk is like a cover of the fruit, fruit and seed sizes, and also the number of seeds per fruit. And what we find is that diet, in terms of attributes of vertebrates, diet is the main, most important variable explaining the contribution to the cophylogenetic signal, being frugivores the strongest contributors, which means animals that eat more than 60% of fruits in their overall diet. Body size was also important, but it worked differently for each group. In the case of bats, it was not important, which are somehow expected since in neotropics, bats do not vary very much in terms of sizes. But for birds, is something that sounds counterintuitive, but smaller birds have greatest contribution than larger birds. But what we think that is happening here is that we have much more species that are small and are mainly frugivores there than larger species that are mainly frugivores. So this might be the explanation here. And in the case of primates, large primates had the greatest contribution, and medium primates and small primates had the same contribution. In the case of plant attributes, we see that fruit color and fruit type are the main variables explaining the contribution. Fruit and seed sizes and number of seeds per fruit were also important, mainly for bats and birds, which is also expected since fruit size and seed size is something that is not a very great constraint for primates since they are able to manipulate different sizes of fruits and seeds. So the second conclusion up until here is that functional attributes of vertebrates and plants determine the strength of the cophylogenetic signal. So this made us think if should interactions then be structured by conserved traits, and as a third step forward, as I said before, the degrees of congruence may be driven either by phylogenetic tracking, host ship selection, and vicariance, which we know that might not be the best explanation, and also by evolutionary conserved traits. So we decided to test if this is true. So to do that, we tested for the presence of phylogenetic signal in functional attributes of vertebrates and plants. And what we see is that vertebrates, in their traits, they have a strong phylogenetic signal in all of them, which made us conclude that, that their interaction niche is very well conserved along the evolutionary time. On the other hand, most traits of plants are very evolutionary labil, which means that plants are very flexible. The only trait that had a significant phylogenetic signal was number of seeds per fruit. So trait similarity among closely related plant species is limited, which might be a consequence of recent adaptative shifts or convergent trait evolution in response to trait lability. And this generates something like an asymmetry of evolution, since we have conserved animals interacting with labil plants. And this made very difficult to detect the presence of the shared evolutionary history, because it is there, but it is there in a diffuse way. So as the main response for my first perspective, if species that co-diversified for millions of years have coupled interaction patterns, as a short answer, we can say that no, because we find a weak phylogenetic signal that arises mainly because labil plants can interact with conserved vertebrates in many ways. So ecological fitting might be more important than deep evolutionary coupling when we try to explain modern interaction patterns. The third step forward, if evolution does not strictly frame network structure, but rather ecological fitting plays a dominant role, how do interactions respond to change? And this takes us to my second perspective, where I look at the ecological responses and I try to understand the potential for networks to reorganize facing threats, basically human threats. So I ask, how do networks reorganize when partners are lost? It is important to keep in mind that interaction structure is not a static thing, as I said at the beginning. So the potential for reorganization is worth analyzing. And then when we started to see what we know about this, we noted that it's something that it's like a field that has not been very well explored up until today. So we decided to dip into the concept of rewiring, which is the ability of a species to form new interactions when partners are lost. Three things about rewiring that it's important to keep in mind is that it is an natural ecological process that arises from spatiotemporal variation in partner availability, which means that it happens regardless of anthropogenic disturbances or not. But despite being possible to happen regardless of anthropogenic disturbances, it offers a pathway for resilience in the face of environmental change and human threats. So to first explore the concept of rewiring, which I had no idea what it was until I started to think about those things, I decided to review the literature and ask, what do we know and how is rewiring being assessed? And I was sad at the end because what I see is that mainly everything that we know about this non-static characteristic of interactions are based on morphological match, co-occurrence, and abundances of interacting partners. However, we started to asking ourselves if it is enough. We know that ecological fitting is central, which are strictly related with morphological matching and so on. But should we look further? Should we look more than this? For example, here, is co-occurrence being assessed in an accurate manner? Here I bring an example. This bird only forages in the canopy. These tree only bear fruits under the canopy. Here, depending on how we see those two groups are co-occurring in space and time, but a chance that this bird to consume this fruit is basically zero. So I'm not sure if what we consider as co-occurrence is the best way. So this is something that stayed in our mind for a while. And other things as well, for example, do nutritional content determine fruit preferences? For example, I have a tree with fruits with high sugar, low lipid, and also a tree with high lipid, low sugar. Those two trees are bearing fruits at the same time, but if I'm a primate, probably I will search for more sugar. And if I'm a bird, I will probably search for more lipid. So it's not the fact that I'm there with those two trees that means that I will interact in the same intensity or the same way with both species. Also, the knowledge about the evolutionary history made us conclude that not all species have the same contribution to the overall cophylogenetic signal, which means that one species might contribute more than others. So we think that the species with strongest shared evolutionary history should be more likely to integrate into the ecosystem and form new interactions, since it's something that is happening along its evolutionary history for a long time. So based on that and other observations, such as individual interspecific variation of fruit traits and other observations, we decided to propose a framework establishing three main groups of factors that we need to take into account when we try to assess the potential for networks to rewire when a human threat or any ecological pressures appear. So the idea here is to integrate these three main overlooked elements that has to do with species importance, interaction importance, and the rewiring rules per se, and propose a more comprehensive approach for predicting how mutualistic networks will respond to current and future environmental changes. I'm not going into deep details in each of these because it's like a world of thing, and we can start asking ourselves for a long time and be here until tomorrow discussing what are important. But I would like to convince you that those three main groups of factors are really important. So to better understand how fruit frugivore networks respond to disturbances, it's very important to know which species are the most important and what happens when those most important species are lost. And have in mind that networks are not just a collection of independent species, but it is an interdependent system where one species depends on the other, that depends on the third species, that depends on the first species again. So it's something that is not static and also it's interdependent. So evidence shows that species extinctions do not happen at random. So keystone species, as we know the most important species in our networks, are central to maintaining the structure and are often the most vulnerable to threats. So when we lose them, the consequences may ripple through the entire system. So take the most famous example that we have in the Atlantic forest in Brazil. Pipil jacutinga and Euterpe edulis. This connection between these two species is something that is very important. Pipil jacutinga is the main seed disperser of Euterpe edulis, but Euterpe edulis is also consumed by other vertebrates. But if jacutinga disappears, the regeneration of the populations of Euterpe edulis will be compromised, and also will be compromised the other species that depend on Euterpe edulis as a resource. So the influence of these keystone species is not only direct, but they shape opportunities for other interactions to attach. So when a keystone species is lost, the system loses also a whole set of possibilities, and it's not just one interaction that is lost. So to understand network resilience and the potential for rewiring, we must go beyond counting species, and we need to start to measure the relative importance of each species. That's what allows us to predict the way, to predict where the system is more fragile and where it has the capacity to adapt. So this takes us to the second element, that is the importance of the interaction. Not as all species are equally important, but not all interactions contribute the same to the cohesion or to the structure of a network. So some interactions are more important, other interactions are simply redundant. So losing one interaction will have a consequence, not the same important consequence than when we lose a pivotal interaction. And a crucial but overlooked aspect of biodiversity loss is what we call interaction extinction. Even before we lose completely one species, we can lose the function that these species make in the system. So another famous examples that we have in the Atlantic forest of Brazil is the defaunation of large-seeded trees consumers. The last megafauna that we have in Brazil are the muriqui and the tapir, and those two species are the main seed disperser of large seeded plants, such as this maybe a sapotacea, and this is a fruit that is very, very related with the tapirs. So we have some places in Brazil where populations of tapirs and muriquis are very small. So the importance here of the role that they do in those places are not effective to maintain plant populations for a long time. So they are there, they are still there, they are not extinct, but the functions that they play is basically extinct in those places. So maintaining a diversity of interactions and not only a diversity of species is fundamental for sustaining community structure. So losing interactions weakens the web of connections, and this also limits the system's ability to rewire. So only by looking at both nodes, which are species, and also links, which are interactions, we can fully understand the robustness of Frugivory networks and their potential to adapt through rewiring, facing either environmental change or natural changes that happen over time. And finally, the third element are the rewiring rules. Rewiring is often seen as something that happens automatically, which means that if I have an ecosystem here and I lose one species, and then at the same time the network will start to rewire, which is not 100% true. Many things can happen, and these species might be not very important for that ecosystem to be structured, and nothing can happen. So it's important to have in mind that this is a dynamic and multifaceted process, and we need to look at all the facets separately. So recognizing this is crucial if we want to predict whether mutualisms will persist or unravel under novel conditions. So to disentangle the new pathways or the new possibilities for those ecosystems, we need to go beyond describing network structure and start to explicitly consider functional outcomes, ecological roles of species, and the context where every interaction is occurring. It is a challenge, a great challenge, to harness the adaptive potential of rewiring, since it is a multifaceted factor. And to do that, we need to understand 4 basic hidden rules, which are not very easy to answer, but we need to try to answer them. That is, which species can reconnect, under what conditions every species may reconnect, by which mechanisms each species is able to reconnect, and with what consequences those reconnections happen. So reframing the idea that we have of rewiring as a simple result of morphological matching, co-occurrence, and species abundances need to be changed by now. So it is an urgent step if we want to anticipate and manage the ecological transformations of the Anthropocene. So from the second perspective, I would like you to take the idea here, the take-home message, that rewiring is not just a theoretical curiosity, but it is a direct consequence for conservation. So we need to start rethinking rewiring now. And if a species persistence depends not only on protecting current interactions, but also on enabling new interactions to occur, then conservation must shift from a static view and transform this into a dynamic perspective, considering promoting redundancy between interactions, enhancing landscape connectivity to give more opportunities to new interactions to occur, and monitoring interaction turnover as an early sign of ecosystem reorganization. So I would like to thank all my collaborators. Without them, this work would not be possible. And also my home university, UNESP, the Lab Lacavi, financial support from CNPq, Brazilian government, Marie Curie, and also FAPESP. And thank you. I'm really happy to answer questions.
Thank you, Lika, for an interesting seminar. Now it's time for questions, so please raise your hands. Yeah.
Hi, thank you for such an interesting seminar and giving us such a perspective with these the changes of the Anthropocene, which are huge. So I have a question, because a lot of your work seems theoretical, and I'm sure that it's based on field data too, you know, either past or present, so that you can see these changes. But one thing I was wondering is, how do you get around understanding the interactions when you're trying to figure out the phylogenetic signals and the congruence? How do you get around interactions that are specific to certain localities? For example, you might have a plant species that has a very broad distribution, and the vertebrate that is very important in seed dispersion or in consuming this in herbivory does not. So how do you keep that in track so that you can understand the congruence?
Yeah, this is actually something that we are not able to control, but all the data that I have for the analysis of phylogenetic signal are empirical observations in the field. So I have the whole vertebrate, the whole list of vertebrates and the whole list of plants interacting in neotropics from many different papers, and then I put this all together, and then I consider every interaction that was observed sometime as one interaction. But it's true that some interactions are really rare, so we have like a threshold I think this interaction need to be present at least in 3 papers to be considered as a real interaction.
And it's done in a geographic framework so that you know where this interaction occurs.
Yes,
Okay.
yes.
I see. Thank you very much.
Yeah.
Thank you. More questions? Okay, I'm going to ask a question myself. I was wondering, because you've been studying the cophylogenetic signal between plants and vertebrates, I guess this is with binary networks. I was wondering if this would make sense to do it with weighted networks and whether it's possible.
It's not only possible, but it's the desire. Since with this weight in the network, you can give like weight for how much the strength of this contribution is also modulated by the importance of the interaction for both plates. So it's something that we need to start doing.
But the data is not there yet.
Not there yet, yeah. Because that's it. What I have in hands is a lot of papers which worked with Frugivory and seed dispersal in the whole neotropics. And then I have the traits of plants and vertebrates, but I don't know how much those interactions happened in each site. For some of them, I do have this information, but then I do not have, as neotropics are very large, I do not have the information enough to include this into the analysis, but this would be like the ideal. So the idea is, in fact, to start to test for the presence of cophylogenetic signal locally, not only regionally as we did for the Atlantic forest, the Amazon and Mesoamerican forest, but locally. Like in one place, we know that those vertebrates interact with this intensity with those plant species, and then test for the phylogenetic signal there.
As a follow-up, has this been done in other systems, like temperate systems, and do they find the same patterns of weak cophylogenetic signal and so on?
It's something that is not very tested. So, yeah.
I was wondering, about the importance of historical data. Where do you, since you're interested in understanding resilience, and you're interested in understanding the response to global change, for example, of these species and these networks, where do you draw the line? I mean, you're just commenting that you inform your work on papers that have been published, and you get the idea about these interactions that have taken place in different geographical areas. But when do you draw the historical or chronological line to say, okay, this was the interaction in the past, and this is what's occurring now? How do you determine that?
The idea here is to use only modern interactions. So we have interactions from the 90s to nowadays. So we do not have like historical data from fruits that are not here or animals that are not here anymore. So it would be very interested in taking like step forward and seeing how phylogenetic signal behaved in the past. This would be really interesting.
So you start in the 90s and you see how changes have occurred from the 90s to present.
We consider the year of interaction as something important in our models, but we do not take this into account. For example, if there is an interaction in the 90s and this interaction is not there anymore, it's not possible to know, because I won't go there and see if this interaction is still true. So I consider that this 4 years, 30 years, 40 years are like a window of modern interactions.
So how do you measure the change? How do you measure the response? Because that response has to be triggered, right? How do you know that there is a response to something? Because you clearly mentioned that vertebrates have conserved traits, and that plants are more liable. So there's probably, the response is probably more often seen in the plant than in the vertebrate, right?
We have not going with very deep details since now, but this could be a very interesting next step, mainly because we want to start understanding what are deriving the reorganization of the networks. But up until now, what I have in hands is this value of congruence between the two phylogenies, how much every interaction, which includes a bird or a vertebrate and a plant, and how much this single species or the single interaction contributes to the overall signal. So I take traits, attributes, and then relate with this contribution. This is basically what we have done.
Thanks very much. So the argument that evolution isn't important is because you can't find evidence of coevolution.
Yes.
But the plant traits you've said are very labile, by which I guess you mean among closely related species, there's big divergence. So that suggests to me that the plants are evolving very quickly to exploit essentially quite a static, highly constrained, quite boring sort of mammalian system. And the plants are essentially, across their range, they're changing their traits to suit whatever dispersers are available and what their particular social organization is.
Exactly.
So it strikes me that evolution is really important in the rewiring, but it's evolution almost entirely on the plants because they have this quite unique reproductive flexibility. They can become asexual, or they can choose to disperse not by rewarding fruits. So the explanation was ecological infill. Was it called ecological?
Ecological effect.
Yeah. Well, the main cause of rewiring, you said, was it? I've forgotten what the phrase was. Ecological fitting. That's right.
Okay.
But it strikes me that a lot of that ecological fitting is actually the plants evolving very fast.
Yes.
In response to their population of dispersers that they can exploit.
Yeah, I think what is happening, in fact, is that it's an asymmetry of evolution. Plants are evolving really fast, and vertebrates are evolving really slowly. So it is impossible to detect any degree of coevolution here, since plants are here and vertebrates are here. So the evolutionary history is somehow important, but not the best explanation.
But adaptive change in the plants is driving the network structure.
Yeah.
Could you get something from looking across the range, as the first question suggested, looking across the range of a plant that has multiple dispersers?
Yes.
And looking at within species trait variation.
Yeah, yeah, that's a good idea. So, yes, this is something that, yeah.
Because I mean, I just don't know how. I imagine fruit, the number of seeds in fruits and size of fruit and nutritional quality of fruit and color is actually there's lots of genetic variation in those traits.
Yeah.
I imagine.
Actually, today I'm not really into talking about seed dispersal syndromes just because of that. It's impossible for one single plant to be consumed by one single vertebrate. It's something that the vertebrate is there, the plant is there, they are hungry, so they will eat it. And that's it. And for the plant, it's perfect since it has many dispersers, and for the animal it's perfect as well since it has like the nutritional content that they need to live.
But the plant wants to stop the one mammal or one species taking all of the seeds away. It wants to spread its risk, I guess.
Something that I expected to find is that a strict relation between bats and Piperaceae, which is a very common and very well-known system, but it's still there. The cophylogenetic signal raises a little bit for, I think it's like 11.6%, but it's an explanation that it's nothing. So even when we are inside a system that we know that are more specialistic, it's still something that is there, but not the main way of explaining this.
Okay, we have some more time for questions.
Thanks for the talk. I'm not I'm not an expert in networks, so my question may not make lots of sense, but I'm really interested and excited of what you're doing. So it's a follow-up question mainly related to the natural history, and I will add a little bit of the spatial perspective. So is there any chance that we may have a cophylogenetic signal in species that do not actually interact today, but that may be used as an indicator that these species were interacting in the past?
Yes, it makes sense, but to test for that, I would need some data that I don't have that are interactions that occurred in the very past and not today. But this would be interesting to test, like for the difference, how cophylogenetic signal worked before the Anthropocene, for example, and how it's happening now. This is a very interesting question, but I'm not sure if we have enough information to do that.
And I will try to link it with rewiring. Is it possible that we can add these potential interactions in rewiring that are based on spatial autocorrelation?
The idea is exactly that, is to start to put inside of the works that are trying to access rewiring to consider every single factor that might drive interaction structure. So yes. For example, here I propose that how much a species contribute to the cophylogenetic signal to be taken into account into those works of rewiring.
Thank you.
Any more questions? We still have 5 minutes, so no? Okay, well, thank you, Lika, for your talk. And as you know, we're going to have some drinks later outside, so if anybody wants to join, please go ahead. Okay, thanks.
Thank you.